Hey everyone, welcome to part 1 of chapter 5. This chapter is going to be about fluids, electrolytes, acid, and bases. Hopefully you have seen a fair amount of this material already in A&P. And if not, you should have had it in Gen Bio in some form, not necessarily specific to the human body, but definitely pH and the different electrolytes. I wanted to start this chapter off with this figure here, looking at water, because that's going to be very important to what we talk about in this chapter. It's important to know, to understand how important water is to the overall functioning of our body. So, as I mentioned, chapter 5, fluids, electrolytes, acids, and bases. And there actually aren't too many objectives for chapter 5. But as you can see, these first two kind of lengthy. And what we're going to talk about in this short section is just what is going on in terms of fluid moving back and forth between the vasculature and the tissue, and the forces behind that, specifically proteins. So that's going to be objective 1, and then objective 2, looking at the development of edema. And there's just 2 figures that you want to focus on for these 2 objectives. So the first thing to look at is the distribution of body fluids. You've got total body water, and then that is going to be divided into intracellular fluid and extracellular fluid. Intracellular fluid is going to be that fluid that is in the cell, inside of the cell, so it's intracellular. Then you have extracellular fluid. That is going to be interstitial fluid, that's going to be in the tissue. Intravascular fluid, that is inside the vasculature. Now, it's also important to remember what are the principal ions that are directing fluid in the different areas. So potassium and phosphate, principal intracellular ions, and then sodium and chloride or chlorine, chloride, are the principal extracellular ions. The concentration of those different ions in the 2 different compartments are what's going to drive fluid movement. Now, here's a lot on this page. It's a lot of good stuff. And what these 2 figures show are fluid movement between the intracellular fluid, the IC, and the EC, extracellular. And this figure is in the 6th edition textbook. This is the 5th edition textbook. And they're pretty equivalent, but I just figured put both in there. And then on the 4th, or the 5th edition, you also have the figure legend right here. So this figure is very important and will be referred to many times when talking about the various body systems. Cells go through metabolism constantly, and in that process yield different products that can be toxic if not removed from the environment. And then filtration is the process by which wastes get removed and nutrients get into the cells. So movement, as you see here, is going to be through osmosis. That is water directing the action. You have a couple of different types of pressure to think about. You have osmotic pressure, which you see here in the interstitial osmotic pressure, intracellular osmotic pressure. That is going to have a pulling effect. All right, osmotic is pulling, hydrostatic is pushing. Now, normally the ICF and the ECF osmotic pressures are equal, so you don't actually have constant movement back and forth or a net movement. There's always movement, but because the ICF and the ECF osmotic pressures are equal, then everything is equilibrated, and you don't have movement into one area but not the other. Now, sodium is the main electrolyte that works in maintaining the extracellular fluid levels, while potassium is the main electrolyte that maintains the intracellular fluid movement. And you can see this cell right here. What's not on your text is looking at the capillary hydrostatic pressure here, and then the capillary oncotic pressure down here. Oncotic pressure is going to be based on protein concentration. Now, albumin is one of the major proteins within the blood that works with maintaining the capillary oncotic pressure. Other proteins, including immunoglobulins and antibodies, also play a role in this, but albumin is going to be the major one. If the vasculature loses proteins, fluid is going to move into the interstitial space because the oncotic capillary pressure goes down and then doesn't work to help draw the fluid in. All right, so osmotic pressure, oncotic pressure are going to be pulling pressure. All right, and you've got that right here. You have a very high you have a higher capillary oncotic pressure, and it's going to cause reabsorption. Filtration, you think about filtering, and it's this entire process, but the fluid moving back into the venule is going to be classified as reabsorption. All right, so spend some time on this figure. Think about it in your mind in terms of the capillary here, the vasculature, and you've got the tissue here. You also have the cell, but we're really going to focus more on the vasculature and then the tissue. All right, this figure is old. All right, it is a figure from a previous edition, and I think even a slightly different version of the textbook, but same author. Some students like to see the numbers to see how filtration movement of fluid occurs when the different pressures fluctuate. At the arterial end here, there is a positive value, which means fluid moves out. All right, if you keep track of where all these numbers are coming from here, you have net filtration pressure of plus 10. That will allow pressure to move out, or fluid to move out. At the arterial end, or at the at the venous end, down here, the pressure is going to be negative, as you see here with this -8. Excuse me. When the pressure is negative, the fluid is going to move back into the vasculature. So I'm not expecting you to understand the value or to know the values, but rather the points, all right, the different types of pressure. If you look at the numbers in the capillary at the arterial end, there would be more fluid content, all right? The pressure is higher, the fluid content is higher because fluid hasn't moved out yet. So there's more hydrostatic pressure, 35 here, than there is at the venous end, 17 millimeters mercury. All right. If you look at the oncotic pressure, all right, as long as the capillaries are functioning properly and the vasculature or the endothelium is functioning properly, all of the proteins that are inside this capillary should stay there. So you shouldn't have a change in the oncotic pressure. All right, so filtration is going to occur at the arterial end because you have a high pressure here, or positive pressure. And reabsorption is going to occur at the venous end. Why? Because you have a negative pressure, a net filtration pressure that is minus, and so fluid is going to move back in. Now, trying to figure out these different arrows here in terms of the oncotic pressure, the hydrostatic pressure. All right, the hydrostatic pressure in the tissue is 2. Hydrostatic pressure in the capillaries 37, so the 37 beats the 2, and it moves out. All right, so oncotic pressure here, 0 versus 25. The oncotic pressure is going to pull fluid back in. All right, also spend some time on this figure, not necessarily memorizing the numbers, but just understanding how the pressure changes because of fluid content and then what's going to occur at each end, the arterial end versus the venous end. Alright, I like this figure. I try not to say this is my favorite figure because then everything's my favorite. This figure shows the mechanisms of edema and how that forms. It's figure 5.2. The point of this figure is to show the development of edema, which is in the middle of the figure in the yellow box. I can't tell that it's yellow, but that's the color in the textbook. So edema is the accumulation of fluid within the interstitial space, so in the tissue. So how does it develop? It develops with the activities that you see in purple, which is hard to see on my computer, but if you look in your textbook, these two guys are purple, and then you have this green colored type thing and this blue color. All right. All of that leads to fluid moving into the tissue. So you have decreased synthesis of plasma proteins. This can be due to liver disease, malnutrition, increased loss of plasma proteins, increased plasma sodium levels, water retention, loss of plasma proteins that can move into the interstitial space. You have decreased transport of capillary filtered proteins. Fluid movement into the tissue. That is what's going to cause edema. All right, so what this means is that there is a fluid distribution problem. All right, not necessarily an excess fluid. That might be a common understanding or a common misnomer. Right, you've got fluid in your legs. You have severe edema. You've got too much fluid. It's not too much fluid, it's just the fluid is in the wrong space. And you can actually experience dehydration during edema because the fluid moves out where it's from a place that's actually usable into a place where it's not going to be used at all. So then, what causes or how does the issue of edema develop? That tells you what, or that leads to the point of what you need to know. Right, and what you need to know, according to Objective 2, is what is bolded, and that's going to be in the pink boxes. So, if there is a question on the quiz or exam, say to list the 5 mechanisms that lead to edema, you would want to be able to write the 5 items that are in bold. And for increased capillary permeability, that is one of the mechanisms that leads to edema. Why does it lead to edema? Because the capillary permeability is an issue, and that causes the plasma proteins to move into the interstitial space. Then your vascular oncotic pressure goes down, and you can no longer pull fluid back into the vasculature. This is caused, or can be caused, by burns and inflammation, but burns and inflammation aren't the mechanism for edema formation. They are the cause. You have a burn and you have inflammation that causes the capillary permeability to increase. So the correct answer for that question, list the mechanisms, increase capillary permeability. Burns or inflammation is not the answer. Now, you may still find that type of a question, but it'll be hopefully more clear. So then, the other issues, increased tissue oncotic pressure, the lymph obstruction, increased capillary hydrostatic pressure, that's going to cause fluid to move out faster, decreased capillary oncotic pressure. All right, so you see that decreased synthesis of plasma proteins. That means you simply don't have enough protein in your capillary to have a high oncotic pressure to pull the fluid back in. All right, and then this is kind of, kind of a gross picture to a small extent. It's called pitting edema.